Hello, my name is Ruben Mesa, and I'm the Executive Director of the Mays Cancer Center at UT Health San Antonio MD Anderson. And it's really a great pleasure to be being able to participate in this MPN Horizon meeting. I'm very grateful for the organizers for having invited me. I've had the great pleasure of participating in this meeting in the past. Love the interaction, the collegiality, and the collaboration between many of the MPN advocacy groups from around the world. So delighted to play a role in this year's meeting and look forward to hopefully a time soon where we can gather in person. Now, they'd asked me to touch base on the clinical trial landscape for MPNs in 2020. Here are my disclosures. So the good news is that there is a very broad and deep pipeline of therapies in development for myeloproliferative neoplasms. And as we've all come to learn during this time of the pandemic, clinical trials are essential for evaluating the safety and effectiveness of anything we do in medicine, whether it's a vaccine, whether it's a therapy. So part of a goal of a clinical trial is how do we improve on where we stand currently with medical therapy. So where are we now? Well, we are able to control increases in blood counts with medicines like hydroxyurea. We've had JAK inhibitors that have made a significant impact on myelofibrosis, polycythemia vera, some with ET, reducing symptom burden, splenomegaly, myeloproliferation, maybe prolonging survival in those with advanced disease. We have interferons that have impact on a variety of levels, including potentially altering the clonal nature of the disease. We have therapies that have helped with anemia, such as erythropoietins, immunomodulatory drugs, and androgens. We even have therapies such as stem cell transplantation. Just ahead, we have many therapies that are in development. We have agents that might help anemia that might become available approved, like luspatercept. We have new JAK inhibitors that we'll discuss that might improve anemia as well as splenomegaly symptoms like momelotinib, improve low platelet count like pacritinib. And now we have fedratinib in the United States, not yet approved in other countries, but likely to come in the near future. Down the road, we have therapies that might impact fibrosis, might further impact the clonal nature of the disease, might further impact the symptom burden, or a variety of different targets to further control the disease. In the future, who knows? Maybe we'll have ability to alter the genetics of the disease through CRISPR, use new immune-based therapies like CAR T, or even vaccine-based therapies. Now, as we think about new therapies, we first have to think about where is our current standard. In PVERA, we know, and these are the US-based guidelines, the NCCN guidelines, that in low-risk patients, we're looking to adequately control the risk of blood clots and bleeding, control cardiovascular risk, and if individuals are not doing well with phlebotomy and aspirin, to then consider moving on to cytoreductive therapy. High-risk individuals, we start with cytoreductive therapy, either Hydrea or interferon as frontline, with Roxalidinid as second-line therapy for individuals who are not responding well. Now, as we think about new therapies in PV and ET, we have to be mindful that people that have these diseases have a very heterogeneous nature of burden. Depending upon how long they've had the disease, the change in counts tend to be high toward the beginning of the disease, but decrease as time goes by. Symptoms can evolve. More headaches, itching, and count-related symptoms toward the beginning. More features indicative of myelofibrosis with night sweats, weight loss, spleen-related symptoms as the disease progresses. Indeed, the issue of time is important as we think about how long someone has had an MPN. Risk of vascular events, likewise, is present but evolves during time. In PVERA, there remain real questions. We have both Hydrea and interferon, which is the optimal frontline therapy. How do we prevent disease progression? Should we be using JAK inhibition earlier? Now, with interferons, there is much discussion. There is both pegylated interferon alpha 2A that in randomized studies has showed at least equivalency with hydroxyurea for controlling symptoms and blood counts. And these the MPN-RC studies that I and many others were involved with. We now have ropegylated interferon alpha 2B, both from AOP in Europe and Pharmacentia in other parts of the world. It now has an EU indication in PV. There's some new data from the low-risk study in PV. It's given every 2 weeks. We don't have data yet comparing these drugs head-to-head, but the major advantage is that ropegylated interferon is being developed specifically for MPN patients. So hopefully we'll both be registered and an option, and I think likely to impact PV and in an upcoming study in ET, a study being led by Dr. Verstavchik and I. Now, the continuing data supporting the use of ropegylated interferon in PV that is being discussed at this meeting was ropegylated interferon versus hydroxyurea in high-risk PV, showing at least equivalency of spleen or of control in the counts and risk of thrombosis and safety throughout the first 12 months of the therapy. Where the therapies tend to then differentiate, and we have advantages for ropegylated interferon, is over time with a continuation PV study. So the longer you're on therapy, the more likely you are to continue to benefit. Next, we have new therapies in the mix that are being looked at in ET. So ropegylated interferon, good data in PV, a new phase 3 study starting in ET, being led by Dr. Verstavchik and I. There's now the LSD1 inhibitor from Imago that has an impact on activated megakaryocytes, with a study in myelofibrosis we'll discuss, but also studies in essential thrombocythemia. Open both at my institution as an investigator-initiated study, as well as in parallel a company study, with a goal of, yes, controlling platelets in people that have failed hydroxyurea, but hopefully having a deeper impact on many aspects of the disease, including fibrosis, symptoms, and clonal burden. Indeed, LSD1 inhibition has a variety of impact on the biology of the disease that may impact both the clonal nature of the disease as well as the cytokines. This is the name of the drug, IMG 7289, and is also being looked at in myelofibrosis with upcoming data at ASH showing improvements in splenomegaly and symptoms in individuals that had failed Roxalidinid. The ET study, there is now a broader US-based study in addition to the one in mine. There's also sites in the UK, EU, Australia, and New Zealand in high-risk ET patients, I should say, that have previously failed hydroxyurea. Now, other important agents in this pipeline in PV, there's an interesting group of drugs called the Hepsidine Agonist. One of them is from Protagonist. This helps to mimic the impact of iron deficiency, but people do not have to be iron deficient. So the hepcidin mimicking the anemia of chronic disease, lowering the hemoglobin, but allowing hopefully the iron levels to improve. So this may broaden the control or deepen the control in individuals that are not tolerating phlebotomy well or tolerating iron deficiency, and give an alternative option to cytoreduction. There's also Givinostat, which has had activity in ongoing studies, as well as the AMDN2 inhibitors, both from Kartos and Idasanutlin, the study that I was involved with. Now let's pivot to myelofibrosis. Many of the therapies are tested in myelofibrosis first because of the more advanced nature of the disease. After we have an accurate assessment of the diagnosis, survival, and our treatment plan, patients may be considered for transplant, and there are transplant studies which remain. Frontline medical therapy, a backup salvage transplant, second-line medical therapy, or therapy toward acute leukemia if that is appropriate. Now in 2020, in most parts of the world, Roxalidinid has become the standard. So many have asked, what does it mean to fail ruxolitinib? And I like to share that failing ruxolitinib depends. It depends on really what are your alternative options, as well as asking the question, has the patient had an adequate dose of the medication or a sufficient time to try to see if it's beneficial? There are many different pathways that potentially are going to be inhibited, trying to impact the biology of myeloproliferative neoplasms. And I'm going to highlight a few of these that are in the clinical trials. First, I would highlight the other JAK inhibitors. We have pacritinib, which is now in the Pacifica study, trying to seek approval, in particular for individuals with the myelodepleted phenotype or individuals with marked thrombocytopenia, low platelets. And we have momelotinib as a second-line study for those that have failed ruxolitinib and have anemia, seeking to improve splenomegaly symptoms and anemia in the MOMENTUM study. We also have fedratinib, and I'll go through some of this data now, approved in the United States, both in the front-line and second-line setting. Now, the Pacifica study from CTI, patients with a platelet count of less than 50,000, pretty advanced disease here, as you see by eligibility criteria, randomized between pacritinib or physician's choice of therapy, and looking at overall impact. The MOMENTUM study, which is now accruing, international study, momelotinib plus placebo versus danazol plus placebo. It's a blinded study. And then individuals after 24 weeks are able to go on to open label momelotinib. For those in your groups who potentially would benefit from either of these studies, I would highly encourage them. They're both good drugs. They both have been well tested and are both trying to seek that final step, both for approval, but also can be a great resource for patients. Now, there are many agents in development for myelofibrosis. Some after patients have failed ruxolitinib, like the LSD1 inhibitor, bomedemstat, others to be added on in addition, others that have been looked at in both settings, such as CPI 0610, the BET inhibitor. There are some that we are using in combination with JAK inhibition, some that I have been involved with myself, the addition of luspatercept, which can help with anemia, addition of hypomethylating agents, addition of danazol, which sometimes can help. There are a variety of agents that are trying to be helpful to deepen spleen and symptom response. There are many ongoing phase 3 studies at the current time for myelofibrosis. One that I would highlight is there's the MANIFEST study, and now subsequent phase 3 studies that are ongoing, but either as monotherapy of CPI 0610, a BET inhibitor having impact against the disease, for individuals that have failed ruxolitinib. Second, as an add-on to ruxolitinib for individuals with a suboptimal response. Or third, individuals that are naive or have not had a JAK inhibitor before in combination. So looking at all three scenarios. Imetelstat is launching a phase 3. It's previously shown benefits that are suggestive of an improvement in survival. And again, it is a trial looking at overall survival as the primary endpoint, led by my good friend, Dr. John Mascarenhas. Now, we know when individuals have come off ruxolitinib, life expectancy is clearly diminished. So it is an incredibly important goal to develop better therapies, particularly if they've had clonal evolution. Some have asked, should we just stop ruxolitinib and have a holiday and resume the medication? That can have some benefit, but alternative approaches or a clinical trial would be preferable. Now, fedratinib, I've mentioned, is available in the United States. Let me highlight some of that data with you. This is an updated analysis from the second-line study of the Jakarta-2 study, where we used more modern interpretation or stricter criteria of what it means to have failed ruxolitinib. What we reported was a solid 30-ish response rate in spleen and symptoms in individuals that clearly had failed ruxolitinib prior to starting fedratinib. It was also tolerated. There could be some GI toxicities that are manageable, and one needs to be mindful of thiamine because of a rare reports of Wernicke's encephalopathy. I had mentioned the pacritinib study before, but here showing the data from prior studies showing good impact on spleen and symptoms in individuals, including those with marked decreases in the platelet count. Momelotinib we have previously shown in the SIMPLIFY study, good improvement in spleen and symptoms and improvement in anemia and platelet count, or at least stabilization of the hemoglobin and platelet count, both showing the benefit of why we have those new phase 3 studies. Now, in myelofibrosis, you could see this very robust pipeline of therapies, some of which I've mentioned, really looking at different aspects of treating the biology of the disease, with the cell cycle checkpoint going after fibrosis, signaling tyrosine kinase inhibitors, additional JAK inhibitors, apoptosis, including the MDM2 inhibitors, which we had mentioned in polycythemia vera, immunomodulatory drugs, and a range of drugs really working on the epigenetics of the disease. This showing some of the prior data from the MANIFEST study and from CPI 0610, impacting cells and their differentiation. And this is in the prior data of the earlier study, showing improvements in spleen and symptoms and patient global impression of change when added on to ruxolitinib in patients who were relapsed or refractory to ruxolitinib by itself and were transfusion dependent. And these were in the non-transfusion dependent patients. Very encouraging results, smaller numbers of patients, so clearly the larger study is necessary. This showing very robust response rates when used in the upfront setting. Toxicities can be a drop in the platelet count or anemia and GI side effects. Second drug I would highlight, it's having kind of a parallel development of adding navitoclax, which works against apoptosis, with ruxolitinib, and here showing improvement in splenomegaly when added on as a second therapy to ruxolitinib. Here showing improvement in the symptoms for these individuals, and the toxicities looking similar to the other agents. You can have a drop in the counts, you can have some GI side effects. This does seem to be a recurring theme in terms of toxicities. Luspatercept, another drug I wish to highlight, it's now available in both MDS and hemoglobinopathies. It's been looked at either on those with anemia who are receiving Rux as well as independent of Rux. And what you see here in the light blue, nice benefits in terms of responses, especially in the non-transfusion dependent patients who were also receiving ruxolitinib. So I do see a situation where luspatercept could hopefully become approved soon to help improve anemia in MF, either with or without ruxolitinib. So where might these fit in our treatment guidelines? Well, in the updated US-based guidelines, fedratinib is considered an alternative frontline therapy to ruxolitinib in the frontline setting, or clearly can be used in the second line setting. Other JAK inhibitors in the second line setting, momelotinib, pacritinib, or the addition of CPI 0610, LSG 7289, navitoclax, etc. Luspatercept or momelotinib may be used more upfront for patients with anemia, pacritinib for individuals with thrombocytopenia even from the get-go. CPI 0610 might have an impact of being a second therapy in that frontline setting. So clearly I've shown you data, slides, and analysis from many friends and colleagues, trials I've both been involved with, but also those that I have not. I'm grateful for the opportunity to have been able to present these things to you, as well as the ongoing efforts of my broader group of colleagues working in the MPN Quality of Life Study Group, both at my center in San Antonio and many close colleagues. And with that, I thank you for the invitation and look forward to the question and answer session.